Hello everyone, welcome to sqadia.com. Today we are discussing the cellular energetics and after have been discussed the process of generation of we have discussed how the electron transport chain plays an important role in the production of the uh, energy in terms of moving the potential, in terms of moving the hydrogen ions from inside to outside against the concentration gradient. We are at the second step in which we are going to discuss the generation of the proton motive force. It means that protons which had been initially moved from inside to outside against the concentration gradient, it's time for those protons to move down their concentration gradient with the help of ATP synthase through the two channels that are present in F2 unit, F naught units, down resulting in the movement of a particular subunit of F1 molecules, which would ultimately yield ATP molecules. So let's get started. So we are going to start off with the discussion of the multiprotein complex in the respiratory chain. That is basically a continuation of the previous changes in the redox potential and the free energy. This would give a bit much more insight to what is going on in terms of the electrons that are moving from one step to another and how protons are being moved with utilizing the energy loss. So we are then going to move towards the discussion of the cytochrome C and Q cycle, and then we are going to start off with the discussion of the structure and function of ATP synthase. So starting off with the multiprotein complexes in the respiratory system, what we have over here, we have the matrix as shown in here, and we have the intermembrane space over here. So what we are going to see in this slide is the movement of the protons against their concentration gradient. So they are going to move from matrix to outside by the utilization of NADH molecule over here. So NADH co-Q reductase, which is complex 1 that we studied in the previous section, plays an important role. Flavin mononucleotide, where the blue lines over here shows the movement of electrons. So they are going to be moved from FMN to the iron sulfur complex that is being catered by the CoQHQ. Now this CoQHQ would be catering these towards the CoQHQ cytochrome C reductase complex, which is the complex 3. Over here, it's important to consider that these are, first of all, the 4 protons moved from inside to the outside, and in this case of the complex 3, 2 protons are moving from inside to outside, right? Furthermore, as the electrons are moving down from complex 3 to complex 4, which is basically termed as the cytochrome C oxidase complex, through the action of the cytochrome C, the electrons are being transported from one complex to another, where they are moving down, further losing their free energy and increasing their oxidation potential. In the redox potential, what happens, they are going to transfer these 2 electrons to the protons along with the molecule of oxygen, resulting in the yielding of oxygen as well. So what happened, 2 electrons that were initially yielded resulted in the formation of movement of 10 protons, 4 from here, 2 from here, and 2 from here, 8 and 2 from, of course, here. So complex 1 collectively transported 4 protons from inside to outside. The complex 3 further transported 2 from here and 2 from here as well. So 4 protons from inside to outside against their concentration gradient. Same goes for the complex 4, which basically transported 2 protons against their concentration gradient to the intermembrane space. The succinate CoQ reductase complex, what happens in this case, succinate is being converted to the fumarate. We are talking about not the NADH, but we are talking about FADH2 over here. So what happens in this case, they are going to yield 2 protons, and these 2 protons are going to move outside with the help of adenine dinucleotide, moving down the concentration gradient of the electron in the electron transport chain, the same process that occurs as in this particular case. So talking about the cytochrome C and Q cycle, we have the cytochrome C. We are at basically the discussion zooming in this particular portion, cytochrome C and C portion. So what we have over here what we have over here is the cytochrome C. It is and this diamond portion that is shown in the orange reddish part is the heme group. So these are going to basically cater the electrons, and these electrons are then going to move down their gradient, resulting in the, as shown over here, these electrons were moving down from the copper to the cytochrome A to the cytochrome A3 complexes down. So it similarly, it is going to move through the complex 2 and complex 3, that of course caters, and this would result in the movement of 4 protons from the matrix to the intermembrane space, as we saw the movement of the proton that are moving from the interspace to outside. So this cytochrome C would ultimately result in the movement of electrons down, resulting in the production of 2 molecules of water, when 2 molecules of water are being produced in exchange for the net movement of 4 protons that are moved outside to the intermembrane space. Talking about the Q cycle, what happens in this case, the co-QHQ is going to be moved to the Q naught side, and it carries 2 electrons. The co-Q is going to move at this Qi side, and of course it does not carry any kind of electron. So what happens in this case, when co-QHQ is moved into this particular side, at this side which is specific for the co-QHQ molecule, it is going to transfer one of the electron to the iron sulfur carrier molecule, which then transfers to the cytochrome 1, and this would be converted back to the cytochrome C. Now this would of course yield 2 of the protons simultaneously as well, resulting in the formation of co-Q. Right, 2 protons were released after 1 electron has been given to the iron sulfur, and 1 has been given to the Bl and Bh, of course, as well. So the second electron of this 2 electron complex from the co-QHQ will go down to reach to the Qi complex. And now this Qi at this particular stage is going to bind the co-Q over here, which would ultimately yield 2 electrons with the addition of 2 protons as well. So at side Q naught, what happens at side Q naught, this particular side, we are seeing that 2 molecules of co-QHQ, 2 molecules of cytochrome C3 positive would yield 2 molecules of co-Q, 2 molecules of cytochrome 2 positive, resulting in the net 2 movement of the electrons, of course, and 4 protons. At Qi side, what we have, the co-Q side, what we have over here, 2 electrons along with the 2 protons are going to bind, resulting in the formation of co-QHQ, and as shown by the step 10. So the net Q cycle, some of the reactions of what is basically going on in the Q naught, this particular portion, what is going on in this particular portion, what we have over here. The 2 protons along with 2 electrons are given in, and 4 electrons are moved out along with the 2 electrons as well, which basically would in detail would ultimately move and would be converted into a molecule of water. So what happens, to summarize the Q cycle, 2 electrons that are going to be transferred resulted in the production of the 4 proton molecules that are released into the periplasmic intermembrane space. This can also be seen through this figure, where 2 molecules or 2 electrons are being moved into from the cytochrome C, and 4 protons are moved out. Talking about the structure and the function of ATP synthase, so it is being divided into 2 major portions. So one is, of course, the F0 region, and second is the F1 region. So F0 region contains 3 substrate, the component A, which is a single subunit, 2 subunits of B, and 6 up 10 subunits of C molecules as well. And of course, the oranges dots, the oranges color show that they are static, and the green one shows that they rotate. So what happens when we are talking about F1 portion, it contains 3 subunits of the alpha and 3 beta subunits, along with the gamma subunits and epsilon subunit as well. So what happens is proton half channel, they have 2 half channel protons in the A subunit of the F0. So what happens, the protons are going to move through the proton half channel and is going to bind with the aspartate 61 portion. This would result in the movement of the proton and through this particular section of the F naught, the C subunit. This would, of course, because it can rotate, it would this F naught subunit is binded with the F1 subunit through the gamma subunit. It is going to cause the rotation of the beta subunit, and because the beta subunit is being attached with the alpha alpha and the rest of the beta gamma subunit, it is going to rotate in a homodynamic manner, resulting in the production of ATP. So when this have been rotated, this proton half channel, which was initially binded, it is going to rotate, it is going to be rotated, and it is going to get entered into the second proton half channel. And when it enters into this channel, there is a conformational change which basically causes the detachment of the proton from the aspartate 61, and the proton is going to be released back into the cytosolic medium. So there would be net movement of proton from the exoplasmic medium to the cytosolic medium, and this would be down their concentration gradient, of course. So in this section, we focused on the multi-protein complex in the respiratory chain, and then we followed our explanation discussing about the case of the cytochrome C and the Q cycle, where we discussed two major portions and discussed what is the net outcome of the two electrons, that is the movement of 4 protons against their concentration gradient. And then we studied the structure and function of ATP synthase, of how protons are going to move through the proton half channel, and they are going to cause the rotation of F naught channel, which would ultimately cause the rotation of F1 channel, resulting in the production of ATP. And after they have been produced, proton would enter into the second proton half channel and would move down the concentration gradient from the periplasmic membrane into the matrix. So that was all for this section. Thank you for watching sqadia.com. I'll see you in the next section of the video.